Okay, so here I'm going to describe some of the interventions that take place downstream of the mu receptor. As was discussed before, there are several steps that occur or can occur after the mu agonist, such as morphine, has bound to the receptor. Um, Here I'm just going to focus on each intervention on its own individually um, to try and outline how specifically they do work and the promise they have. The first thing I'm going to talk about are GRK2 and 3. So GRKs are a group of kinases which phosphorylate the mu receptor. Now, kinases are simply the term used to describe proteins which carry out phosphorylation. And phosphorylation itself is simply the addition of a chemical group called a phosphoryl group to a particular protein or enzyme. Phosphorylation is important and occurs throughout biology and throughout the cells and different interactions within our bodies to change the behavior, change the activity of the protein to be phosphorylated or dephosphorylated. In the case of mu-opioid signaling, phosphorylation is crucial to determine the behavior of mu receptors, which in turn has important behavioral consequences. For example, the phosphorylation state can determine how fast the receptor becomes desensitized, which can lead to tolerance. It can increase the rate of recycling of the receptor, which can decrease tolerance as more receptors that can react or respond to the mu agonist are returned to the surface. In the case of GRK2 and GRK3, the phosphorylation of the mu receptor occurs at serine threonine amino acid residues found on the C terminal. We use the term C terminal to describe the part of the transmembrane protein that ends in a carboxyl group, which is the COOH. As opposed to the C terminal, you have the N terminus, which faces on the outside of the cell and is contains a NH2 group. So the relevance of GRK2 and 3 here is because through the introduction of this phosphorylation, the mu receptor becomes more likely to be desensitized, meaning it's more likely to become decoupled from this G protein signaling we see here, and ultimately can no longer lead to the effects of analgesia or euphoria. It's also important because it's been seen that this phosphorylation by GRK2 can also affect how quickly the mu receptors are returned to the surface as well. Indeed, there is evidence to show that by inhibiting GRK2, the degree of tolerance that builds up in preclinical models is reduced. And this might be due to a combination of reduced internalization and increased recycling, so increased movement to the surface.